Hello, my name is Miss Day, and I'll be reviewing meiosis with you today. So, first of all, what is meiosis? Hopefully most of you remember, but in case you don't, meiosis is when we take one parent cell and we make 4 haploid daughter cells out of it. Hopefully you remember what haploid means, but in case you forgot, haploid means we have half the number of chromosomes in these 4 cells that are made during meiosis. These 4 cells also have a special name. They are called gametes. In humans and many other animals, these gametes are known as egg in females or sperm in males. In plants, these gametes can take the form of plant spores. Now, why do we have meiosis anyway? Why do we have this in life? So meiosis is the reason we have variation. It is the reason your brother and your sister, or a sister and another sister, all look different from each other. They're the reason we have genetic variation. Just as a note, meiosis happens in all sexually reproducing eukaryotes, so it happens in plants, animals, and fungi. Alright, now that you remember what meiosis is, let's talk about where meiosis happens. So, does meiosis happen in all cells? No. Hopefully you answered no to that. But meiosis only occurs in cells that can create gametes. So in humans, for example, the only cells that can form gametes are in females, ovaries, which create the gametes, eggs, and inside males we have testes, which create the gametes, sperm. All of our other cells in the body are called somatic cells. These somatic cells undergo mitosis, not meiosis. So, let's review a little bit. Somatic cells. Hair. Is our hair made out of somatic cells? Alright, before we move on to what exactly the steps of meiosis are, we will be reviewing some key terms. First of all, let's look at the picture on the screen. This is a karyotype of a person. That just means that's a picture of their condensed chromosomes lined up nicely for you. Over here we can see that chromosome 3, for example, we have 2 of chromosome 3. They're both identical to one another, and therefore we call them homologous chromosomes. This means that we can point to the chromosome 3 on the right and say the homologous chromosome of this is the chromosome right next to it. Now, on this karyotype, you can notice that there are 23 pairs of homologous chromosomes that look alike in size and centromere position. So, humans have 46 chromosomes, but we only have 23 pairs of homologous chromosomes. Now, where did each of these homologous chromosomes come from? Well, if we look over here, let's say at chromosome 15, the chromosome on the left may have come from your mother during meiosis, and the one on the right may have come from your father during meiosis. Each pair of chromosomes has one chromosome that you inherited from your mom and the other chromosome that you inherited from dad. Before meiosis, we have something called interphase. During interphase, S phase specifically, the chromosomes replicate themselves to prepare themselves for either mitosis or meiosis. When chromosomes replicate, they become 2 identical sister chromatids that are attached at a centromere. The X shape that you see is called a chromosome, while the 2 sides are called chromatids. Okay, now we've officially gotten to the steps of meiosis. You've heard the acronym PMAP before, which stands for prophase, metaphase, anaphase, and telophase. Well, this acronym can be very useful to help you remember the steps of meiosis. P for prophase. The chromosomes are P, preparing for division. M for metaphase. The chromosomes are lined up in the middle. A for anaphase, the chromosomes are going apart from one another, and T for telophase, the cell is tearing apart. Now we have to remember that in meiosis we do PMAT twice. In meiosis 1 we do prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis. Then we go ahead and do meiosis 2 where we do prophase 2, metaphase 2, anaphase 2, telophase 2, and then again cytokinesis 2. Now let's start out meiosis 1 by looking at the first step, prophase 1. In this step, the nucleus dissolves and chromosomes pair up to form tetrads. In tetrads, there are two chromatid pairs joined together at their centromeres. Tetra means four, so hopefully tetrad will be easy for you to remember because in a tetrad, there are four sister chromatids. Prophase 1 also has a very special event called crossing over. This is where homologous chromosomes exchange pieces to make new gene combinations. They do this when they are joined together in tetrads. This crossing over event between homologous chromosomes is the whole reason that meiosis provides genetic diversity. Next, we have metaphase 1. In this stage of meiosis 1, the tetrads line up along the middle of the cell. Spindle fibers are also reaching out and attaching to the homologous chromosomes at the centromere during this stage. In anaphase 1, sister chromatids remain attached to one another and move to opposite poles because the spindle fibers pull them to these opposite poles. In this case, the homologous chromosomes are separating. In our last stage of meiosis, our cell tears apart to form two cells, and we have now a haploid set of DNA in each of our two cells. We're now moving on to meiosis 2. In prophase 2, the chromatid pairs are released from the nuclei of the two cells once more as the nuclear envelope dissolves. In metaphase 2, we have our chromosomes lining up in the middle of the cell. The spindle fibers attach the centromeres in order to prepare for pulling them apart during anaphase 2. In anaphase 2, we now have what used to be sister chromatids but now are called individual chromosomes moving toward opposite poles of the cell. They are being pulled by spindle fibers to these opposite poles. As a note, this is the first time that individual chromosomes even exist inside the cell since interphase, way back before even meiosis 1. Alright, now we're finally at telophase 2 and cytokinesis. This is the final stage of meiosis. Hooray! This is when nuclei form as the cells begin to separate out from one another. So the end result of meiosis 1 was two cells, and each of these cells formed two new cells during this telophase 2 and cytokinesis. Alright, we did it! We're almost at the end, guys. Now let's just go over meiosis one more time, and I also have a picture of mitosis on the screen for comparison. In meiosis, we start off with this parent cell. It is not a somatic cell. It is either an ovary cell or a testes cell in humans. It's going to form an egg cell or a sperm cell at the end. This egg or sperm cell is going to be haploid. As you can see, haploid means it will have half the number of chromosomes as the parent cell. Okay, remember, meiosis is PMAT twice. So in meiosis, we do prophase, metaphase, anaphase, and telophase twice. You can remember this by P, we're preparing for meiosis. M, the chromosomes are lining up in the middle of the cell. A, anaphase, the chromosomes are being pulled apart. And T, telophase, the cells are tearing apart and forming new cells.